my name is Taylor Moldenhauer. I'm a second-year grad student, bio track, and behind the camera is Alex Bright, a second-year grad student, chem track. We today are going to give you a little virtual tour of our lovely Grace E. Harris Hall and the Forensic Science Department. So grab your virtual mask and let's go.
Alright everyone, so here we are on the second floor of Harris Hall, and we're going into the Forensic Science Department now. What we're going to see on our left are all of our lovely labs, and some are bio, some are trace, some are chem through this hallway. Some of the most important features about this hallway and our Forensic Science Department that you will see coming up are the TA office, where you can get help from any of our grad students, and Joe's office, where you can get help with, just general grad student things. You'll see our lovely posters. You'll see the lovely information about our department and how great our faculty have done. So here we have our first little office with Mackenzie in here. This is where you'll find your TAs. Usually we'll have a lovely little door with all of our TAs showing who they are, so you never forget a name. Say hi, Alex.
Hi.
Here we have Dr. Cruz's office, as well as many of the other offices of our faculty in here. So if you need to come to office hours or just say hi, that's where they'll be. Moving on, we have Joe's office. And then finally wrapping around to the other side of our hallway, we have all of our teaching labs, where you will take all of your awesome science-based courses.
So this is our crime scene room, where we have all of our crime scene classes, both regular and advanced. It's a fully equipped crime scene lab, so we can go through stuff like forensic photography for crime scenes. We even have a really fancy 360-degree scanner to analyze the crime scene room. We have a super glue fume chamber for fingerprints. Someone is actually doing research with that right now. And we have a whole bunch of mannequins over here because at the middle and the end of every semester we do mock crime scenes, which is basically us TAs come up with a crime and you guys have to solve it in class.
Alright, so moving on, we've now entered our bio labs. So we're fully set up to do appropriate social distancing, as you can see with our dividers here. Don't mind our mess. We also have all of our microscopes out so that we can properly spread students out right now. So we're a little cluttered. That's okay. So VCU has some excellent instruments that are really fundamental to DNA forensic casework, and you get an excellent education here because you have such great hands-on experience with a lot of these instruments. Something to point out is our Thermo Cycler over here. We are fully equipped to do PCR, not to mention there are multiple labs with more updated versions of this that if you get into the more advanced labs, you will have experience working with. We've got two very large fume hoods, as well as a little smaller one that we can keep everything nice and uncontaminated with. We also have a QIAGEN QIAcube over there to do DNA extraction work with. So really, as you can see, we're very set up. Another feature of our biolabs is that we do mock cases. We work off of, the TAs will set up, a sexual assault case for you that you then work with a partner through. That gives you really good experience in learning how to not only go through a case from serology to the end where you're writing up a case report. So that's pretty awesome.
So this is our microscopy room. We have all of the microscopes set up for fire, debris, and explosives, the forensic microscopy class, which everyone has to take because these are fundamental instruments for every single track. We all use them constantly, as well as the firearm and tool mark class, where you guys use this fancy comparison microscope to look at cartridge cases, bullets, and tool marks. In the microscopy class, where you guys learn how to use the two main ones, we have the regular compound microscope and the stereo microscope. You guys will be analyzing a whole bunch of stuff like hairs, lamp filaments, which is a common thing with car accidents and stuff. You also look at paint, polymers and whatnot. What else? I guess pause it.
All right, so moving on from our lovely labs, just wanted to show you all our closer look at forensic science board, where it shows all of our awesome grad students and PhD students and postdocs working very hard, and all of the labs that we have. Look how cute we are. Look how happy we are, even in COVID. No, let's do the collab.
So now we are on the 3rd floor, which is kind of like the little end of the forensic science department. And
See, you can notice that by the crime scene tape, obviously.
yes. We have Dr. Miller and Dr. Simmons' offices over here. We also have a handy dandy collab room.
Only for grad
Only
students
for grad students.
and teachers.
Which is, let me turn on the light. We have like a little table for you guys, so you guys can sit. We have a fridge, so we can keep our food in here.
We like that.
And yeah.
And, not to mention, look at this lovely wall of alumni. Look at how happy they are. Think about the awesome jobs that they have found, because VCU is the program to be in. Look, that's our class. There we are. All right.
So yeah, during not COVID times, we all hung out up here and just had a great time. We all talked. We all do schoolwork up here, but right now there's a social distancing, so only a couple people can be up here at once. But we also have a whole bunch of other stuff at VCU that's useful for grad students. There is a grad section in the library that is only available to faculty and grad students, and it's a silent floor, and it's fantastic. There's a whole bunch of resources up there that grad students can use. The VCU Forensic Science Department is very centrally located, so we have the Commons where you can go get food right across the street.
It's where the big ram horns are.
Yes. We're really close to both parking garages, so you guys don't have to walk far if you pay for a parking pass. We're really close to the gym, and we're near a whole bunch of restaurants, so that's really nice.
Yeah, we gotta give a shout out to Little Mexico.
Yeah.
Yep, that's where you go after instrumentation. Also, keep that in mind. We all go to Little Mexico. Well, when it's not COVID times. But I mean, so obviously, as you can tell, we've got a lot of great resources here up at VCU, meant specifically for grad students to help everyone out with that.
Yeah.
All right, so moving on.
Instrumentation Suite.
Instrumentation Suite.
On our way to the Instrumentation Suite, we have this lab, which is used for the wet lab portion of the toxicology class and instrumentation. We share it with the chemistry department, so there's a whole bunch of stuff around. And that's Jessica, one of the other lovely TAs.
Yay!
That is on the chem track. So this is the handy dandy Instrumentation Suite, which we can use for any classes that need any of these instruments research. We also share this with the chemistry department. But there's so many things in here that are useful to the forensics department. We have a couple of the IR, infrared spectroscopy machine instruments. We have a couple Ramens. We have a whole bunch of GCMSs and a couple of HPLCs. The DART isn't in here, but we have a DART for the toxicologists and the controlled substances students. We even have a really fancy laser cutter that we use for Dr. Simmons' research for cutting teeth, which is really cool. And we also have an ICP.
All right, so we're now finally making our way to Temple Hall official to see the lab that I work in, to give you an up-close view. But I just wanted to point out, as we mentioned, we've got the Commons right across here, another lovely bridge, so all of our departments are technically connected, don't even need to walk outside, which is pretty nifty when it gets colder out here. And so yeah, here's a lovely view of our campus as we go to the Earhart lab.
Oh, and that is the gym right there.
Yes.
So it's really close.
Yes,
And that is
amazing.
the life sciences building?
Yes, life sciences, you may have a couple classes
Yep.
in this little building here. Unfortunately, it's not connected. All right, so one more stop before we hit up the lab. We have the offices of Dr. Earhart and Dr. Alvish in here, as well as another big classroom that you will most likely be in for your instrumentation if things come back online. All right, so now we're inside the lab of Dr. Earhart, where I do a lot of my research. We are fully equipped to work with BSL-2 microbes, and what he's got going on right now are avirulent, so non-pathogenic, non-harmful strains of anthrax, as well as Yersinia pestis. So we are equipped to handle those kinds of microbes. He does a lot of microbial forensic not casework, but like lab work as well. We have a full GC that I can show you. We are set up to run PCR on samples, as well as do quick qPCR with our fun little Chi system over there. Please excuse our mess. We got a lot going on in here. We got a lot of different projects. I think there's at least 6 people working on different research projects right now. So like I mentioned, we've got a GC in here, which is pretty cool. We combine chemistry and biology all in one. So yeah, we got a lot going on.
All right, everyone, so that concludes our short little virtual tour of our department. We hope it's really helped you see our campus. It is a beautiful one, and even so, with COVID going on, we're definitely being safe up here. I think VCU is one of the better campuses that has really tackled the issues that are going on right now, and hopefully things will be a little bit different for you if you decide to come to VCU.
Yeah, and I hope the rest of the open house is a good time for you guys and answers all of your questions. Makes this school a priority.
Yep, so have a good one.
Bye.
Hi, welcome to the lab for Forensic Toxicology Research here at VCU. I'm just going to give you a brief tour around the lab so you can see all of the instruments that we are fortunate enough to work with. So this is our Gcms. We use it for pretty much all kinds of screening methods. We look at e-liquids to determine their chemical constituents. We've evaluated real-life samples that people have sent in after poisonings. Over this way, we have Headspace GC dual FID or FID-MS. It can go either way. So one of our master's students is currently looking at both methods to determine the pros and cons between them for blood alcohol content analysis. Down this way we have another Headspace GC dual FID, which another master's student has been working on evaluating alcohols and other volatiles in e-liquids. And then over here we have an LCMS, which we've used for all kinds of analysis purposes, such as quantitating cannabinoids and plant material or e-liquids themselves. So in the lab, as a student, you get to work with all of these instruments. It's very hands-on, fantastic skill builder. So we hope to see you soon, and let us know if you have any questions.
Welcome to the Forensic Anthropology Lab at VCU, Department of Forensic Sciences. The Forensic Anthropology Lab has all kinds of skeletal material, and we also conduct research and physical evidence in terms of the duration and quality of latent prints on lit and unlit light bulbs. We have references. There are numerous casts showing trauma and pathology, and also a fairly large collection of 12 fetuses of known age and sex, and these are real skeletons. We have all the equipment necessary to conduct forensic anthropological fieldwork and casework. We have more skeletal material, all of which is real. We have fragmentary skeletal material for students to work on as well, in terms of being able to identify things to body part, side, and anatomical feature. In the past, we've conducted research on blunt force trauma. We also have things like thermal imaging cameras, slow motion cameras to capture trauma taking place. We have a very nice Leica microscope in order to do work on cases, and also a Faxitron box x-ray machine, again for casework and trauma analysis and experimentation. In the past, we have also conducted quite a lot of work on trace isotope analysis in surface and bulk dental enamel in order to help identify the origin of individuals who are deceased and have remained unidentified.
Hey guys, welcome to the Sing Lab. So the Sing Lab is a molecular research lab here at VCU, but instead of looking at human DNA evidence like what is classically associated with forensic science, we are actually here to investigate the insects and the microbes associated with crime scenes. So you can see we have a pretty traditional DNA extraction setup. We have a couple of flow hoods here. We also have stations set up that are going to allow us to identify those forensically relevant insects. A lot of the research here in the Sing Lab does deal with the decomposition process. So for this, we've had large field studies that really allowed us to, survey this timeline of decomposition, which is going to, allow us to make better inferences about some crime. We also were able to get insect and microbial evidence. This is really important because we need to see how those specifically impact that decomposition process. Now, that's not the only thing you can do with microbes. We also have a couple of projects ongoing here that are using bacterial signatures to identify forensically relevant body fluid samples. Now, to do this, we do use what's called massively parallel sequencing. This is basically a function where we're able to put DNA in machines, hundreds of samples at one time, get our results relatively quickly and very confidently. So for that, we do have an iron torrent and more recently acquired the MiSeq FGX. Thanks for joining me.
Hey, welcome to the Dawson Cruz Forensic DNA Lab.
We mainly try to focus on developing ways to better process mixture samples or samples that could have either different cell types in them or cells from different contributors. This is what we term our pre-side laboratory, so anything before we amplify or make a lot of copies of the DNA, we perform over here. One of the projects that we're focusing on now tries to take antibodies that are targeted to specific cells and use those to pull away and separate certain cells from other cell types. This right here is one of our micro device platforms to help us automate that process.
Hi, I'm Sarah. I'm a second year master's student. So I'm working on the chip project that Brittany just mentioned, and my part is to develop a extraction technique that works on the currently used antibody binding method and that contains small volumes so that it'll fit on the chip. And you can see one of our samples here.
And now we'll go over to the second side of our laboratory, which is anything after we amplify multiple copies of DNA.
I'm Chastin, a second year PhD student, and my research is focused on developing a mixture detection assay to be used at the corn step that will be used on evidence items to determine if they contain DNA from one person or from multiple people.
Over here we have a lot of different instruments, including instruments that Chastin uses for her high-resolution melt curve analysis. Here is another instrument that we use to determine the amount of DNA within our samples. And finally, our genetic analyzer, which after we've made multiple copies of the DNA and tagged certain loci or